Well, good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Establishing Efficacy with Flow Cytometry-Based Receptor Occupancy Assays. My name is Sumaya, and it's my pleasure to be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, you can contact me anytime by sending a message using this chat panel. We also have two handouts for this presentation, so audience members, you can download them through the Handouts tab in your control panel. At this time, all participants are in listen-only mode. Please also note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Medpace who developed the content for this presentation. Medpace is a scientifically driven, global, full-service clinical contract research organization providing phase 1 to 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. Medpace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and disciplined operating approach that leverages local regulatory and deep therapeutic expertise across all major areas, including oncology, cardiology, metabolic disease, endocrinology, central nervous system, and antiviral and anti-infective. Now, it's my pleasure to introduce our speakers for today's event, and our first speaker is Priscilla Bressler. Dr. Bressler is a PhD scientist with specialized experience in immunology and analytical assay development using flow cytometry. She has extensive experience in flow cytometry, investigating the biology of neutrophils in patients and innate lymphoid cells on rodents. In addition, she has experience in drug discovery in the field of neuroinflammation using iPSC-derived microglial cells. Dr. Bressler is well-versed in laboratory operations, ensuring accurate, precise, and medically reliable data. She has also participated in research and development within the flow cytometry area with direct oversight of assay validation and implementation. Our second speaker is Andre Olson. Dr. Olson is a senior scientist who has 14 years experience in hematological flow cytometry. He has a decade's worth of experience serving in a hematology division in a top US hospital with demonstrated publications and solid knowledge in myeloid differentiation and single-cell RNA-seq omics. In addition, he has a deep understanding of big data in clinical research. He earned his PhD in experimental hematology. So now, without further ado, I'd like to pass control to our speakers. So Andre, whenever you're ready, you can go ahead and get started.
Thank you for that nice introduction. I have shared my screen and I hope that everyone can see it.
That looks great, Andre.
Thank you. So today the topic is establishing efficacy with flow cytometry-based receptor occupancy assays. So in order to understand receptor occupancy, we have to talk a little bit about flow cytometry, because flow cytometry is the technology that is used to perform receptor occupancy assays. So I will start off by talking a little bit about how flow cytometry works and how it can be applied in a clinical lab. Then Priscilla Bressler will talk about the definition of receptor occupancy, as well as how the design of an assay can have an effect on its function. And then if we are to implement a receptor occupancy assay in a clinical laboratory, what is required of the assay? And then lastly, I will look a little bit, or I will talk a little bit about the benefits of receptor occupancy assays in clinical trial. Is PD modeling possible, for instance? So flow cytometry, how does it work? So if we break down the words, it's a pretty good way of understanding how it works. So flow in this context means fluid. Cyto means cell. So it's cells in fluid. So in flow cytometry, you arrange the cells with hydrodynamic focusing, which then forms a steady stream of single cells. These cells can be queried by a light source, and you can measure the transmitted fluorescence. Um, and once you've done that, you can measure it. But if we look at the transmitted light, the forward scatter is a representation of the size of the cell. So the larger distance in the forward side is forward scatter, the larger the cell. The reflected side scatter is a reflection of the complexity of the cell. So a denser, more complex cells will reflect more light to the side. And just these two properties are correlated to each other. So using light scatter forward and light scatter side, you can profile cells based on their size and complexity. So just a simple explanation like that shows the principle of flow cytometry. You have fluidics that is necessary to align the cells. You have optics and detectors that are necessary to profile the cells. But then one of the strong aspects of flow cytometry is the electronics, because you can measure thousands of cells per second. And if you have the processing power to deconvolute that, you have an enormous statistical basis for your analysis. And that creates a high throughput multiplex single cell resolution instrument. But of course, we've asked ourselves if we can do better than size and complexity. And to showcase that, I show a cell here with different surface proteins. These can be receptors or ligands or structural proteins. But if we have a cell and we think of these surface proteins as antigens, we can develop antibodies that are specific for this receptor, but not necessarily this receptor. So the antibody enables us to identify a cell. And if we then have a different cell that, say by microscopy, looks the same, but we find that there is a different antigen on that cell, we can develop a different antibody that's specific to that. So the antibodies enable us to separate these two different cells. But the cytometer cannot detect antibodies. So how do we overcome this obstacle? Well, fluorochromes are small molecules that have spectral properties that enables them to be excited by a specific wavelength and then emit light at a different specific wavelength. So shown here, it's the fluorochrome Efficacy that gets excited by 488 nanometers and emits its peak emission around 540. So over the time, different fluorochromes have been developed that can be specifically detected by different filter sets. So the same light source can excite and create specific light patterns for these three fluorochromes. And in the flow cytometer, you have a spectral setup that enables this detection. But in the case of our example with the two cells that look similar but in fact are different, we can take these fluorochromes and conjugate them to our specific antibodies and then label the cells. And how that looks on the cytometer then is that you will have the single positive label cell type B show up in this dot plot, and those cells are different from the single positive labeled cells that have the antibody A. So just with two antibodies alone, you can identify 4 different cell populations. So quite powerful just through that simple setup. So if we then look a little bit on how this can be applied in the clinical lab. So over time, different markers have been developed that, or I should say different antibodies have been developed that identify different markers that are specifically expressed on certain cells. And today, there's something about 400 different commercially available antibodies. So you have an enormous power in combining antibodies to find the cell type of interest. So with that, combinations of markers can lead to greater cell specificity. And so what we see here is if we just use one antibody, we can separate out all leukocyte groups from erythrocytes. If we add a second antibody, such as CD3, we can separate out all T cell lymphocytes from other cells, such as B cells, NK cells, neutrophils, etc. If we then add a fourth antibody, we can separate out all T helper cells from cytotoxic T cells and other cells that we haven't labeled for. So just with the addition of a couple of 3-4 antibodies, we can look at T cell subsets. And if we then combine that with the forward and side scatter that I talked about early, we can subgate on the lymphocytes or gate on the monocytes. So just with simple means, we have the capacity to look at specific cells. So in a clinical lab, the main power of a cytometer is its precision and accuracy. So in a clinical lab, we can look at heterogeneous samples such as blood or bone marrow or any fluid that has cells in it, and we can specifically identify cells within that heterogeneous sample. And with that, we can do a relative quantification, so we can say how many % of cell type A is there to cell type B. We can do an absolute cell count by cell enumeration. So for instance, one in vitro diagnostic assay that we run profiles the CD4 and CD8 T cells, and that's useful in HIV tracking. We can profile hematologic disease processes by labeling for various markers. So for instance, we can track B-cell malignancies over time by using B-cell markers. We can use stem and progenitor cell markers and track myeloid malignancies over the course of a study. We can use cell sorting to isolate a specific cell type of interest if we want to look at that downstream with technologies such as PCR or single-cell RNA-seq. So in a nutshell, that is how flow cytometry works. And flow cytometry is a technology that has been in the making for some 40 years. And in the start, you had one light source, a laser, and you detected 1 to 2 different fluorochromes. Today, there are commercial instruments that have up to 9 laser sources and can profile up to 50 different parameters at any given time. So flow cytometry has developed enormously in the last couple of decades. With that, Priscilla Bressler will talk about receptor occupancy.
Providing a definition of receptor occupancy. So Andre, you can go to the next slide. So we can define receptor occupancy as multiple measurements that demonstrate the binding of a therapeutic that we can call the drug to its extracellular target that we can call the receptor. So usually these drugs are antibody-based therapeutics, and they target specific receptors to modulate the signalization pathway downstream. So we can go to the next slide. Andre, we can go to the next slide. All right, so I just want to illustrate a little bit here with this example. So here you can see a natural killer cell that is expressing a NCR, natural cytotoxic receptor, that we can call NKp44. So when the ligand will bind to this receptor, a signalization pathway will be activated and then will induce the production of pro-inflammatory soluble molecules, and these molecules will contribute to the inflammation and the disease. So if we go to the next slide. So the objective for the sponsor will be to target NKp44 receptor, and the expected downstream effect will be to decrease then the production of these inflammatory molecules, and then to decrease the inflammation and the disease symptoms. So for that, the idea will be to use a monoclonal antibody that will be specific to an NKp44 epitope. So by performing a receptor occupancy assay, we will be able to know what is the percentage of receptor occupied by the drug. We will be able to know if this drug is efficient and specific, or what will be the mechanism of action of this drug. So to reply to this question, we can use the flow cytometry technique. And at MRL, we have experience performing flow cytometry assays like receptor occupancy. And receptor occupancy assays are among the most difficult assays to develop. And I hope this webinar will help you to provide some insights to better understand and design correctly your receptor occupancy assay. Let's go to the next slide. So as Andre explained earlier, flow cytometry is a powerful technique because we can measure the target engagement on a specific cell within an heterogeneous cell population. However, we can face some challenges. For example, you can have a low percentage of cells expressing the targeted receptor in your cell suspension, and having a rare population can be challenging to reach the sensitivity of your assay. So if we go to the next slide. So when the receptor occupancy can be used. So we can use receptor occupancy in preclinical toxicology studies in order to confirm the target engagement in xenogenic species. We can use as well receptor occupancy in PD, pharmacodynamic measurements, and PK, pharmacokinetic, to optimize clinical trial design and to guide the dose selection. So this is actually what we are doing at Medpace with several sponsors, and Andre will discuss this further in the last part of this webinar. So receptor occupancy as well can be used to assess the impact of neutralizing anti-drug antibodies, ADA, on receptor engagement, because the issue with this ADA is that they can inactivate the effect of the therapeutic. So when we are performing a receptor occupancy assay, we will be able to know the mechanism of action of the drug, so if this mechanism of action is maintained or not. So now in the next slides, I will provide you some information regarding the different types of measurements that we can perform to address the percentage of receptor occupancy in clinical trials. So here, the first measurement that we can do is the percentage of free receptor. And we are doing this measurement to determine the percentage of receptors that are not occupied by the drug. So this percentage of free measurement is important while assessing the blocking capacity of an antibody. So here, you want to block the interaction between the receptor and his ligand, and you want to block the downstream signalization pathway. So this is exactly what the example I explained just before with the NK cells that produce inflammatory molecules. So while performing a free receptor assay, we can use two types of antibodies. We can use either a competitive antibody or a fluorescently labeled drug. And in both cases, we will work in conditions where the sample is in saturated condition. So while we are saturating this sample ex vivo with the unconjugated drug. So first, let's say that we are using a competitive antibody. So the competitive antibody is a fluorescently labeled antibody, and they have the specificity to compete to bind to the same epitope of the unconjugated drug. So if the competitive antibody is used, it's really important to make sure that the specificity of this antibody is equal or lower than the therapeutic drug. Otherwise, the competitive antibody will displace the unconjugated drug, and then you will underestimate the calculation of the free receptor occupancy. So if we go to the next slide, I will explain you the second possibility to calculate the percentage of free receptor, and for that we can use fluorescently labeled drugs. So this is the easiest solution actually, because you don't have any competition, and in that case we are still working in saturated conditions, and the fluorescently labeled drug will directly bind to the targeted receptor in the same way as the unconjugated one. So while using either the competitive or the competitive antibody or the fluorescently labeled drug, you will target only the receptors that were not binded by the unconjugated drug. And the signal that you will obtain, so the percentage of free receptors, will be incorporated in the equation to calculate the degree of receptor occupancy. So this was for the percentage of free receptor. So if we go to the next slide, I will explain you the second type of measurement that we can do. So this is the percentage of bound receptor. And this assay is used to measure the percentage of receptors bound by the antibody drug. So this approach is used when antibodies to the receptor are not readily available, or for example, if you don't have a really good specific signal. So how it works? So in that case, you can use a secondary anti-drug fluorescent antibody, and this antibody will bind to the drug, which is binded to the receptor of interest. So in that case, the percentage of bounded receptor can be used as well to calculate the percentage of receptor occupancy. And usually this assay is combined with the total receptor measurements, and I will explain you this one in the next slide. So for the total receptor measurement, we measure the totality of targeted receptors independent of the presence or absence of the drug. So this will provide some insight into the level of receptor internalization resulting from the drug antibody binding. So the percentage of total is measured that time using a non-competitive antibody. And this non-competitive antibody will bind to a different site of the antibody drug on the same receptor, so that you will be able to measure the totality of the receptors in the surface of the cell. So we usually, you combine this assay with the bound receptor assay that uses a secondary anti-drug antibody. So this combination enables the measurement of the total receptor expression on cells. All right, so we can go to the next slide just to summarize a little bit because it was a lot of information. So in conclusion, when you are doing a receptor occupancy assay, you are able to evaluate the percentage of free, the percentage of bound, and the percentage of total receptors on their own. And you can use as well these three in combination. So for example, you can use the percentage of free in combination with the percentage of total, the percentage of bound with the percentage of total. So the percentage, in the first case, it will be more an indirect method, and in the second case, it will be more a direct method. So you can see in the bottom of the slide two types of equation that we can use for receptor occupancy estimation. And for each receptor occupancy assay, you will have a specific receptor occupancy equation, and you will need to choose the correct one, and this will be the part of the development of your assay. So now we can go to the next slide. So now let's talk a little bit about how to design and optimize a receptor occupancy assay. So the first step with any assay development is to understand the context of use of the data. So this is mostly the part of the sponsor investigation, and the two major points that will need to be considered is the design input and the design process. And these two, design input and process, are really important not only for receptor occupancy, but for all flow assay that you want to design. So the first step during the design input is to understand the biology of the cellular populations of interest. So we can address some questions. So for example, what is the phenotype of the cell population? How can we characterize these cells? Are they lymphocytes? Are they non-hematopoietic cells? What is the potential of activation of these cells? Can we find them in a different states in healthy versus disease samples? And the last question that we can assess is what is the localization of this cell population? And this will be very important to determine the specimen matrix to run your assay. Can we find this population in the blood, in tissues, in bone marrow? So these questions are really important and should be addressed before performing a receptor occupancy or any flow assay, for example. Let's go to the next slide. So now we can discuss a little bit about the design process. So in the design process, this part is more where the scientist can work closely in collaboration with the sponsor. So several questions can be raised. So we can, for the panel design, for example, we can really wonder which markers do we want to use, what is the level of expression of the population targeted. So you really need to know that the cellular population selection is really important. So the receptor occupancy measurements at a given concentration of drug can be cell population dependent. So for example, a higher expression level may result in a more robust assay. So this is really important to check before. What about the fluorochromes? So which fluorochrome we want to use for the assay? So if you have a population with a very low expression, so sometimes it's better to use a bright fluorochrome. You can as well work in and trying to understand if you have a good separation between the positive and the negative population in your population. So this is always something that we need to consider when we develop a flow assay. And for that, we can work on optimizing the titration of our antibody reagents, and as well to calculate the staining index. So I just put an example here in that slide. So the staining index is the ratio of the fluorescence intensity of the positive population and the negative population divided by 2 times SD. So the higher staining index you have, the more we can clearly define a positive and a negative population, and the more you will be confident on your results. We can go to the next slides. So let's talk about the antibody choice. So this is really important as we're working in flow cytometry. So once the type of measurement has been selected, you will need as well to identify if you want to use a competitive or non-competitive detection antibody. And this is really important to understand the affinity of the therapeutic agent, because as I explained before, if we choose a monoclonal antibody with a higher affinity, then the drug can induce a displacement of the therapeutic agent, and then we can underestimate the percentage of free receptor. So it's really important to understand this part. We can as well play in the affinity consideration by really playing with the incubation time and temperature and concentration. So for example, if you have a low concentration of an antibody with a low affinity, you may need to increase the time of incubation or in order to increase the chance of detection of the receptor by this antibody. So regarding the saturation, so all receptor occupancy antibodies must be used at a saturating concentration because we really want to bind all the available binding sites. So we really want to find the optimal saturation concentration between as well different cellular concentrations. So for example, we want to compare healthy versus disease samples. And in that case, I think that the minimum of subject that you need to use is a minimum of 3 because you can have donor-to-donor variability. So this is really something important as well, the saturation of your antibody. Okay, we can go to the next slide. So let's talk about the gating strategy. So yeah, we are working again now with flow cytometry. So we really need to target in a specific way our population. So we really need to design a gating strategy in order to optimize the correct resolution of the population of interest. And we need to be as specific as possible. So just some tips, if I can say that. It's really good to include a live dead dye in order to exclude the dead cells, in order just to exclude false positive staining. It's good as well to exclude the doublets, so to have a doublet discrimination dot plot. Including a time gate as well is very useful to control the fluidics consistency during the acquisition. Having a negative control as well is very useful to confirm the positivity of your signal. And as well, something to me that is very important that we should work on at the beginning of the clinical trial is the reportable. So because that can take a bit of time to set up. So which units you want to use? Do you want to use the percentage of parents? Do you want to use absolute count? Do you want to use the mean fluorescence intensity? So this is really something important. How do you want to report your data? Okay, regarding the sample preparation, so once the panel has been designed, the sample preparation must be optimized. So in the case of a method transfer, so the sample preparation protocol usually is already designed by the sponsor, designed and optimized by the sponsor. However, sometimes when you transfer an assay to the clinical lab, some steps could be adjusted if necessary. So this is always good to communicate with the sponsor and with the clinical lab in order to really optimize some steps if necessary. However, if we design from scratch, all the steps should be optimized. The incubation time, the number of washes, and as well all the reagents will need to be chosen in an appropriate way. All right, so we can go to the next slide. So regarding the instrumentation, so we really need to wonder if the instrument will be used with the correct optical configuration in order to fit the desired fluorophores that are included in the panel. So as an example, at MRL, we have 2 instruments, the BDFAX Canto and the BDFAX Lyric. So for any design of receptor occupancy assay, it's always good to discuss with the clinical lab and see if this assay is feasible with the configuration that is in the clinical lab. You really need as well to think about the acquisition capabilities of the instruments, such as the flow rates or the possibility to acquire in tubes or plates, because all instruments are different. Just something that I think is really important when you develop an assay, it's always good to avoid running samples in the high flow rates in order to avoid having a lot of aborted events. So this is something that you need to consider. Regarding the carryover, this is always an experiment that we include at Medpace in our validation plan. So it's an important parameter that we need to consider just to avoid as well carryover in high samples to low samples. So we can go to the next slide. So pre-analytical considerations. So I think that choosing the correct specimen matrix is really important. So the best specimen matrix to use to me is the peripheral whole blood, because it's the most commonly used specimen type for receptor occupancy. And using whole blood is almost not invasive and contains a decent amount of immune cells. However, the selection of the anticoagulant is very important, as this can alter the expression and the stability of the target antigen. Whole blood as well is preferred as PBMCs, because with PBMCs you are doing a processing before using PBMCs. So if you have a bad enrichment, you can have an impact on the receptor occupancy calculation. Again, as well, if your cell population is in low abundance in the circulation in the whole blood, sometimes you can use as well tissues. However, if you are using tissues, you really need to consider that you will need to process these tissues. You will do a mechanical and an enzymatic dissociation, and this can alter the antigen expression and affect the integrity of your tissue residing cells. All right, so that was it for my part. So now I will let Andre discuss about the benefit of receptor occupancy in clinical trials.
Thank you for that introduction, Priscilla, on receptor occupancy. To close out this seminar, in the last part, I will just talk a little bit about how receptor occupancy can be used in clinical trials. So in pharmacodynamics is the study of the biochemical and physiological effects of drugs. So receptor occupancy assessments, as Priscilla has shown, are sufficiently sensitive to monitor target binding and thus are very useful in drug development. Receptor occupancies assays has been developed and applied in both non-clinical and clinical studies to provide insight into PK-PD relationships so far. And also a little bit on what Priscilla said, is like drug target is optimally on the circulating cells, but if you don't have that available and your target is on tissue, you can still use the receptor occupancy assay on circulating cells as a surrogate measure, but it will obviously not be your primary assay. Moreover, we can use receptor occupancy assays to provide valuable insights at all stages of drug development. So initially, we can understand the mechanism of action or the binding characteristics of the therapeutic with a specific target. During phase 1 clinical trials, where dosing protocols are developed, receptor occupancy assays can track and then thus correlate if you have severe side effects, so you know at what receptor occupancy levels that would happen. We can use receptor occupancy assays during the identification of the biological effects using different doses, and then couple that with PK and PD assessments to guide dosing protocols. Then in preparation for phase 2, we can use receptor occupancy assays to help predict levels where the receptor is modulated by the therapeutic. So far, receptor occupancy has been used as a PD marker for a number of therapeutic antibodies, such as anti-PD-1, AMG 479, ATR 107, and etrolizumab. And at Medpace, we have seen an upswing in the receptor occupancies assays that we've been awarded in the last 2 years. 2 of these have made it to phase 2. So next I'm going to talk a little bit about how you can utilize receptor occupancy assays. So here we see data from one of our sponsors where we have measured the receptor occupancy, and then they have analyzed it through the course of the treatment. So we can see initially at screening that we have variation in the different subjects. The receptor occupancy ranges from 0 to about 30%. But you can then see how efficient the drug is, because right at the start of the study, you reach 100% receptor occupancy that is maintained throughout the course of the study. So just a simple plot, it makes you see how the receptor occupancy performs over time. In the next example, MRL produced the receptor occupancy data, and then the biostats at Medpace used the plasma concentration of the drug that had been established by MBL through LCMS, and they did a exposure response analysis using a sigmoid Emax model, where we can see how the receptor occupancy is in a dose-dependent relationship with the drug plasma concentration. So at lower concentration, we have low amounts of receptor occupancy, and as we increase the plasma concentration of the drug, we get a higher percentage of receptor occupancy. This modeling was used to establish the EC50. So these were 2 examples of how we've used, or the sponsors have used, the receptor occupancy we have produced in modeling their data. So in summary, flow cytometry-based receptor occupancy assays, they specifically measure the degree of a drug that is bound to the receptor of interest on the cell type of choice. Along with immunophenotyping assays used for clinical immune monitoring, receptor occupancy assays have become a standard application in flow cytometry in the context of drug development. Receptor occupancy assays are increasingly used in PK/PD modeling and are becoming more and more the recommended way to ensure the effectiveness and the specificity of a therapeutic. Oh, sorry. With that, I would like to thank the webinar attendees for their attention, and I would like to thank the organizer for letting us talk about this topic.
Fantastic. Thank you, Priscilla, and thank you, Andre, for that wonderful presentation. And now that brings us to our Q&A portion. So audience members, you can continue sending in questions for Priscilla and Andre. We've already received some questions, so I'll start off with those. Our first question is stability needed to be considered in RO assay?
Hi, this is Priscilla. So I think that I can reply to this question. So Stability is important not only for receptor occupancy assay, but as well for all flow assay. So at MRL, we perform different types of experiments, so I will not go into too much detail. So we are looking at pre-analytical stability. So we are looking at the specimen matrix stability before staining. So this is really important for the shipping condition, for example. And we are looking as well at process sample stability to look at the stability of your sample after staining, just to know how long I can keep my stain sample at 4 degrees before doing an acquisition on the cytometer, for example. So the stability is simply tested by calculating the percentage of bias of receptor occupancy between a freshly drawn sample, which will be your baseline, and different time points from the same specimen. In the same time as well, you can compare 4 degree and room temperature. Usually we are using a criteria of 20% of bias. If the stability is not found, you can collect the blood, for example, if you are using blood using Streck tubes, and these Streck tubes contains a stabilizer. Then you can push the stability to more than 10 days. However, this can be a bit risky because the stabilizer can impact the extracellular receptor detection and then impact the RO. And you can as well organize with the central lab to analyze samples within stability. So we have an example here at Medpace where the stability was below 12 hours. And in that case, we organized with the lab to have a night shift and to analyze the samples during the night. I hope this replied to your question.
Great, thank you, Priscilla. And our next question from an audience member, and that question is, how does low volume of blood affect the assay?
Okay, I can reply as well. So if we are using, if we draw blood, for example, with a small volume, first, you need to consider that when we are doing flow cytometry, we always need to have a decent amount of blood if we need to repeat an experiment. When we are doing receptor occupancy assays, sometimes we are doing another experiment, which is immunophenotyping in parallel. So you need to have enough blood to repeat if necessary and to do these two assays. Another thing is if you have a low volume, you can really have a poor specimen quality and you will not be able to reach the sensitivity of your assay. However, when you plan your clinical trial, you need to respect an ethical requirement to minimize the potential adverse effects from collecting large blood volume from subjects. So you really need to calculate what is the minimum volume you need to draw for your sample.
Great, thank you, Priscilla. And our next question is, what is the benefit of measuring total receptor?
I can comment on that. So the total receptor is needed as a point of reference. So if we have receptor internalization, for instance, that is reflected in the total receptor. And as Priscilla indicated in the talk, you normalize the free or the bound receptor to the total, so you can get a measurement on the level of receptor occupancy.
Great, thank you so much, Andre. And our next question is, how long does it take to develop RO assays range from conception to validation?
Maybe I can comment on that. So I can reply for Medpace. So here at Medpace, when we develop an assay from scratch, including the optimization, that can take 14 to 16 weeks. But we always need sometimes to include a feasibility experiments just in order to see if we have a decent amount of cells to stain the samples and to have a good sensitivity.
So our next question is, how is the detection method decided in receptor occupancy?
I can comment on that. You can look at your free receptor or your bound receptor, and you can perform accuracy and precision on that and see which one performs the best, and then go with the free receptor if that has greater accuracy, or go with the bound receptor if that has greater accuracy. So it comes down to precision and accuracy.
Great. Thank you, Andre. And our next question is, in the case of an RO assay that uses a secondary antibody and an antibody with a different fluorochrome against a different epitome on the same receptor, how do you relate MFI to number of receptors for the different fluorochromes?
Yeah, so Priscilla briefly touched upon that as well. You can use standardization beads for that particular fluorochrome, which then has a known amount of fluorochromes on them. So you can standardize the MFI on one fluorochrome to the MFI of a different fluorochromes using standardization beads.
Great. Thank you so much, Andre. And our next question is, for RO and whole body, what precautions need to be taken for collection and how soon does it need to be analyzed? Can RO be done on bone marrow, aspirates, and biopsies?
Yeah, so I can comment on that as well. So the main consideration for the matrix is stability and the associated accuracy that comes with it. So if I generalize, different cell types have different stability. And like Priscilla indicated, that can be affected by the temperature and the anticoagulant. But even if you know that one cell type, for instance, T lymphocytes, are stable over time, that doesn't mean that all the receptors on the T cell is equally stable to the general T cell. So you have to assess that in your validation. So in validation, we look at stability of your specific marker of interest, and we make sure that it maintains the accuracy needed over time. And that that's the same for bone marrow, essentially. It's just that bone marrow is a much harder matrix to come by. So with bone marrow, for instance, we might have limiting amount of cells, so it's not the stability that is the issue. It's the fact that we don't have enough cells from the biopsy to ensure accuracy.
Fantastic. Thank you, Andre. And our next question is, can the binding RO data be used to be as a good marker for efficacy? Can it be used to personalize dose for a patient to have more efficacious and safe dose for a patient?
On its own, the receptor occupancy can not be used for efficacy, but coupled with other data, you can definitely customize the dose response of the particular subject, because you get, as soon as you've done your receptor occupancy assays and analyzed it, you have an answer on the level of receptor occupancy assay.
Fantastic. And our next question is, if the target cells are on tissues, but we want to use PBMCs as surrogate, what needs to be done?
Yeah, so I'll comment on that as well. If you have your target cells on any type of cell that is within PBMC, we do the whole validation on the PBMCs, because as Priscilla indicated, when you isolate PBMCs, you process the cells, and that might change how the receptors behave. So if the receptor occupancy assay is established in whole blood, for instance, but because of reasons in the study, it can only be performed on PBMC, we have to perform a validation and establish our criteria, that our criteria is upheld on PBMCs just as they were on whole blood. We can't just assume that it will work on PBMCs because it worked on whole blood.
Fantastic. Thank you, Andre, again. And our final question is, how can RO assays be used to clinically interpret values in clinical studies?
Yeah, so that's a good question. So the receptor occupancy assays has to be correlated to safety data or any type of PK or PD data that is associated with the study. On its own, the receptor occupancy is a number. We can tell whether the drug saturates or it is at non-saturating levels. But in the end, that has to be correlated with safety or other type of data in the study.
Fantastic. Well, thank you very much, Priscilla and Andre, for all of those answers, and for everyone else for sending all of those interesting questions. We've reached the end of the Q&A portion for this webinar, but not to worry, we did receive lots of questions, so not to worry. If you do have more questions that weren't answered, the team at Medpace may follow up with you later on as well. And if you do have further questions, you can also direct them to the email address on your screen. So go ahead and quickly jot that down. So that's info@medpacelab.com. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our webinars. Now, please join us in giving a big thank you again to our fantastic speakers, Priscilla Ressler and Andre Olson, for that wonderful presentation. We hope you found this webinar informative. Have a great day, everyone.